Hey everyone, welcome back. We're going to dig into chapter 12. Chapter 12, if you remember chapter 11, we were talking about ways to control microbial growth. Chapter 12 is really going to be digging into how we do that inside the body. So that's really all it is, talking about drugs, antibiotics. So we're going to talk about the cellular targets of the antimicrobial drug therapies, how they work, and we are going to get a little bit into a little more specific on these than we did on the control chemicals. The structure of common antimicrobial drugs, the chemical structure, I know. The modes of action of each class, the mechanisms of drug resistance microbes have developed, and like how they fight back, and then identify the side effects of antimicrobial drug therapies. So the goal is to administer a drug to an infected person that destroys the infective agent and does not harm host cells at all. But unfortunately, the perfect drug does not exist, not in any way. So we just have to sort of compromise a little bit. So we look for selective toxicity. That's a very important term that is definitely going to come up again. Selective. Oh, for crying out loud, I forgot. Sorry. Selectively toxic. So a big point here that will definitely be on the test is selective toxicity. And what does that mean? Selective toxicity is referring to the ability to kill the microbe, but not harm the host, like toxic to the microbe, non-toxic to the host. That's selective toxicity. It's not toxic to both. So we want something that's microbicidal, that kills, rather than microbistatic, that just slows or stops growth. We want something that's soluble, functions when diluted in body fluids, is potent long enough to act, and is not broken down or excreted before it can be effective. Doesn't lead to the development of antimicrobial resistance. We have that way that it might complement or assist the activity of the host's defenses. Remains active in the host tissues and bodily fluids for a while, readily delivered to the site of infection. Even if it goes all over the place, at least it gets to the site of infection quickly. It's reasonably priced, and it does not disrupt the host's health by causing allergies or predisposing them to other infections, other than being toxic to the host themselves. So that's the goal of what we would like to look for. Terminology that's important in this chapter, we have the term chemotherapeutic. A chemotherapeutic drug is any chemical that is used to treat, relief, or act as a prophylactic for disease. So that means prevent disease. Essentially any drug. Okay. A prophylaxis, like we were saying before, that's the use of a drug to prevent imminent infection of a person at risk. So we'll talk about prophylaxis in the term of infection, but of course you can talk about prophylactics as being like things that prevent, pregnancy. People use that often. So antimicrobial chemotherapy, use of drugs to control infection. Antimicrobials are any drug that targets microbes. That's just a broad term. And an antibiotic. Now it sounds like anti-bio is anti-life, but that is incorrect. This has become, these are substances that are produced by natural metabolic processes of some organisms or created by scientists chemically that can inhibit or destroy microorganisms. I'll say microorganisms, but generally, generally the term is used for drugs that are targeting bacteria and not other types. So when I talk to you guys about treating infections, viral infections, do not spit out at me about antibiotics treating viral infections, because antibiotic is a term for any infective. No, antibiotics typically is a term used even in the medical community to treat bacteria. So keep that in mind. Don't argue with me over it. Okay. more terminology that you might want to know. Semi-synthetic drugs are drugs that are chemically modified in the laboratory after being isolated from natural sources. They're just modified. Synthetic drugs are drugs that are produced entirely by chemical reactions within a laboratory setting. So just totally synthetic, made in a lab. Narrow spectrum or limited spectrum. That means that they work against a limited amount of microbes. So it might be specific to gram-positive bacteria. Okay. Broad spectrum or extended spectrum means these are things that are active against a variety of microbial types that could be both gram-positive and gram-negative bacteria. It might not work against fungus, but it works against a ton of bacteria. That we would consider that broad spectrum. Okay, the antibiotics typically are common metabolic products of bacteria and fungi, just to prevent the growth of competition, right? To get rid of the competition. That's what they're typically for. New drugs are created by chemically altering the structure to make semi-synthetic drugs. I mean, that's what we do. We take the structure, the chemical structure of whatever, like penicillin, and add things onto it synthetically. So that's a semi-synthetic. Synthetic drugs, however, those are going to be the things that we can't get them from any habitat or population or whatever. We can't get to it. Sometimes it could be because we can't get there. Sometimes because of destruction of habitat or whatever, but we try to just mimic what we would see in a laboratory. So before you start somebody on antibiotic therapy, you want to identify the organism. So these are pretty important steps, right? So number one, identify, I don't know what is going on with my lip gloss today. It's just been awful. I've done like 3 videos. It's just all sticky. Right. You want to know the microorganism, you want to know the degree of its susceptibility to drugs, and you want to know its overall, the overall medical condition of the patient. So if the patient has to be cured quicker versus somebody that can wait for the treatment to work longer, if the patient can handle a drug that could be more toxic, or if they can't handle that more toxic drug, those are things you might want to consider. One of the most important tests that we do, that you guys have done in the lab probably by now, is the Kirby-Bauer assay. The Kirby-Bauer assay, you pop some antibiotic discs onto an agar that you've spread a bunch of bacterial growth onto. If the bacteria can grow, then it is not susceptible to that antibiotic. If it does not grow, you'll see a clearing around that disc. It is susceptible to that antibiotic. We can determine drugs that bacteria could be susceptible to in order to give that to a patient, or we can even determine strengths of drugs to use, like how strong do we need to give this vancomycin. This is another test that works very similarly. These strips are the numbers that are on them down here have to do with how concentrated the drug is on the strip. So you can like determine like the growth based on that. It's a similar kind of concept here. This is just like kind of what you guys see with ELISA, but it has to do with a positive control. Positive looks purple. So if there is purple, so this is the point where it stops being effective or stops growing is what I'm trying to say. So sorry, this one was for this one down here. I was looking at the X. It's like you guys could read my mind or something. So we grow. This is what it looks like when the bacteria can grow. We no longer could have growth because we are now susceptible to the antibiotic at this concentration. So we go from, it even says it on here, increasing concentration of the drug from left to right. So what does no zone of inhibition indicate on a Kirby-Bauer disc diffusion assay? If there's no zone of inhibition, so it's a disc, but everything grew, and that bacteria is not susceptible to that drug, right? So let's look through our choices. High toxicity to the patient. No, we can't tell that from Kirby-Bauer. No. Bacterium is resistant to the antibiotic. That might be a way that we could talk about it. The low toxicity to the patient, can't tell that from this. Bacterium is susceptible to the antibiotic, but we said there's no zone, right? The no zone, then it is not susceptible to the antibiotic, so it's resistant to it. Okay, so if your treatment with your antibiotic fails, that failure is due to these possible choices. The inability of the drug to diffuse into that body compartment. That's number one. So it couldn't get to the brain or whatever, like the blood-brain barrier. That's a pretty serious thing that to get a drug across that. Like, I'm not kidding. Like, it is, some drugs just can't cross the blood-brain barrier. So that could be an issue. Joints are sometimes an issue. If it's on the skin, sometimes it's hard to keep antibiotic like saturating the area. Resistant microbes in the infection that didn't make it into the sample collected for testing. So if there are other microbes, like you just took a little swab of this guy's wound, but he has a lot of wound, and where you checked, we didn't grow the resistant microbes, but where there are resistant ones over on the other side of the wound, now they're going to overgrow because we treated it with that. But that's an example. Bite might fail because you can't get a good representation every time, unfortunately, depending on the wound. And then an infection could be caused by more than one pathogen. So it could be mixed, and some of those could be resistant to the drug. So those are all possibilities of why that might have failed. So the therapeutic index is the ratio of the dose of the drug that is toxic to humans to its minimum effective therapeutic dose. So the lowest amount, the ratio of the drug that's toxic to humans. So let's say, whatever the ratio of that drug, penicillin or something is toxic to humans, ratio of that to its minimum effective dose. So if it takes, 2 grams of penicillin to make somebody have a rash, but it takes 1 gram of penicillin to kill the bacteria, right? So you might be able to treat that person because you're below that therapeutic index there. Like the closer you get to 1, like if this was 2, this one here, if this was 2 grams, that is a 1-to-1 ratio. 2 to 2 is 1-to-1. It's the same thing. When you reduce it down. So the closer these two figures are to each other, the smaller the ratio, the greater potential for toxic drug interactions. So the TI of 1.1 is riskier choice than a TI of 10. So the closer it gets to 10, the closer it gets to 1. So anyways, that's the point. That's what I'm trying to say. When drugs have similar reactions, problems with patients or effectiveness, the drug with the highest of this has the widest margin of safety. So that might be the one that you would want to go with. So has similar effectiveness. So just think of it as that way. Okay. Okay. Patient history also has to be considered when you're choosing. We want any pre-existing medical conditions that will influence the activity of the drug or response of the patient. That's hard to be aware of sometimes because sometimes patients aren't as forthcoming as they should be. History of allergy to any certain class of drugs. Some people don't even know that they do have allergies. Not that they may have had one, but they just don't know that's what they were allergic to because they just don't keep track of it. They have underlying liver or kidney disease, also hard to predict. Infants, elderly, and pregnant women are also special precautions. Like if you have a pregnant woman, some of these antibiotics could cause defects, birth defects. So you want to be careful and consider all that when you're choosing. So this is coming back to selective toxicity. We're going to go back to this important topic here. It's the central concept in antibiotic treatment. They should kill or inhibit microbial cells without damaging host tissues. High toxic to bad guy, low toxic to host. So the best drugs in current use will block the actions or synthesis of molecules in microorganisms, but not the vertebrate cells. Yes, attack these guys, but not these guys. So drugs with excellent selective toxicity, we can start with the penicillins. They block synthesis of the cell wall that's found only in bacteria. So that's not going to affect us, our body at all. Drugs that are most toxic to human cells are ones that act upon structures that are common to both bacteria and us, like DNA or something like that. So characteristics of infectious agent become more similar to that of the host. The selective toxicity becomes more difficult to control. We're especially talking about viruses whenever they are hijacking our own machinery, right? So we want to target anything that is different in the pathogen compared to ourselves. So metabolic targets of chemotherapeutic agents include the cell wall, synthesis of it, inhibiting the nucleic acid crap, the protein synthesis itself, yeah. And then we have folic acid. So this is a new one that we didn't have on the other one, right? And then interference with the cell membrane. So we had these ones. We had, this was always 1, this one was 2, this one was 3, and this one was 4 on the last one. Now we're introducing a 5th one. So these are the 5 ways that these can work. So you might want to be aware of those. That when we're talking about drug action, stick on that folic acid synthesis onto the end of how the other ones worked in the previous chapter. So these are all the different ways that drugs can act on bacteria and how bacteria function. I'm not going to go through all of these right here because we're about to go through all of these in the next few slides. But this is a great way, a great summary for you guys to look at when you guys are studying. And even like do like pin the, you know, drug on the bacteria or something. That'd be a great way to study for that. Yeah, I mean, I don't know, similar sort of chart. Yeah. Here's some drugs, and if you want to see, you know, which ones can work on what, like tetracyclines, look, they could all these up here. So, example. You can look at it yourselves. Anyways, these are the penicillins. These are anything that's related to penicillins, and these all have something called a beta-lactam ring. Very important to know that. This is like the defining feature of penicillins is the beta-lactam ring. And then they also have this thing called thiazolidine, but you might like be able to recognize that. I'm not going to ask you to really like be too familiar with it, but yeah, so these are all related to penicillin. And so we have nafcillin, ticarcillin, cloxacillin, carbenicillin, like all of these are related to penicillin. They're just derivatives of it. Penicillin was naturally derived from mold. And now I guess nowadays we can synthesize it. So it has 3 parts. It has the thiazolidine ring. It has the beta-lactam ring, and then it has a side chain, whatever that little side chain is off of this junk. This is what it's talking about, the R group. So that's the structure of how it works. And what it does is it breaks down these cross bridges here. So it just breaks them apart. So now we have our cell wall falling apart. So what is the mechanism of action of penicillin? Does it disrupt protein? No. Does it block peptidoglycan cross-linking? Does it disrupt the cell membrane? No. Does it inactivate DNA transcription? No. So it was this one. When you guys are going through your questions on your exams, I do recommend that you go through all of the choices, just because some people don't realize that there's like an all of the above. And sometimes the all of the above is like at the bottom, or it's like at C or whatever. So just be sure you're actually looking through it and choosing the one that you mean to choose on your exams. Sorry, it was my husband. Cephalosporins, right? So these also target the cell wall. They also have a beta-lactam ring, kind of similar to penicillins. So we also have carbenipins, similar idea. These are the beta-lactams. We have the penicillins, we have the cephalosporins, we have the carbenipins. I don't need you to know the ones that we have not talked about right now. Why? I don't know. Anyways, moving on. Pretty commonly talked about ones, we have bacitracin. This is a pretty common ingredient in Neosporin, so that affects the cell wall. Then we have like vancomycin. So these also attack the cell wall, but they clearly don't look like a beta-lactam. So beta. These do not have a beta-lactam, but they do target the cell wall. They just work differently. But these are like the three that are separate. Okay, moving on to protein synthesis. The ones that attack protein synthesis are called aminoglycosides. They usually have an amino, sugar, and an amido cyclotol ring. I mean, so they're from actinomyces species like Streptomyces and Micromonospora. Fine. They are broad antimicrobial spectrum. They have some side effects that include a neurotoxicity, allergic reaction, nephrotoxicity, which is your kidneys, and ototoxicity, which is your ears. So this is how drugs that affect protein synthesis will work. There's some different ways. These are all different. I want to be clear. So these are all different. So the these ones, they come in and they inhibit the ribosome from attaching to the mRNA essentially, and the other side of the ribosome. So they inhibit that, they block that. The aminoglycosides, which is what we were just talking about, these come in and inhibit the positioning of the mRNA strand. These guys, I can't read this on here, pluromutulins. I don't know if I need glasses, it's just tiny, it seems so tiny. Anyways, it prevents the formation of peptide bonds is blocked. So what I'm trying to say here is where you would attach your amino acids to, they can't stick on anymore, it blocks that. Tetracyclines, which you've probably heard of, and glycocyclines, similar drugs, will block the tRNA from coming in and matching onto the mRNA position. So you can't make it, you can't make the protein. And then erythromycin prevents the ribosome from moving along down the mRNA. So that's how they all kind of work. I would just be aware that they can function these different ways and that these are protein synthesis blocking drugs. I'm not going to ask you individually necessarily about each of them, but I might ask you about tetracycline. Is it a protein synthesizing drug? But I just mean, I'm not going to ask you like, does it work by binding the blah, blah, blah? I'm not going to ask you that. So tetracyclines, breaking these down. Broad spectrum. I don't know. If you have to know, it's a natural parent compound and a synthetic derivatives related to this. But anyways, broad spectrum, and it has these side effects here, gastrointestinal and just deposition in hard tissues. That includes in your teeth. People often get lines on their teeth when they have to have taken it. It's like a gray line across their teeth. Glycocyclines, it's a derivative. It blocks the ribosome subunit and is effective against things that are resistant to tetracyclines. Moving on to erythromycin. Erythromycin is a macrolide ring. That's the structure that it has. It's broad spectrum, low toxicity. And then we have clindamycin, which is adverse reactions in the GI tract. And then we telithromycin, which we use for respiratory tract infections. That one just drew a line through it. That's right. But you guys got it. All right, moving on to folic acid. Folic acid synthesis. This is a pretty straightforward one. I like this one. Sulfonamides and sulfa drugs. So sulf. Look for sulf. Sulf. Folic. Okay, let's cut it out. Folic acid synthesis. So these are the first modern antimicrobial drugs, and they are synthetic. They don't originate from bacteria or fungi. This includes sulfasoxazole. I'm really bad with like words in general. It's hard for me to talk sometimes. So speaking these words is just excruciating. So sulfadiazine, that's pretty commonly used for burns. Trimethoprim, sulfamexazole. Trimethoprim. Okay, so this is the idea of it, man. I mean, sulfonamides, just they have a sulfur group in them, and that's the important part. So these side groups that are, do you have this squiggly guy coming on? The squiggly guy can attach to any of these guys, and that's what we're talking about over here. So how does it work? essentially whenever bacteria are making folic acid, which is an important precursor to like a lot of stuff, they have their little enzyme, that their little substrate normal thing. This is a normal function here in square number 1. That's normally what it does, right? The sulfa comes in, it's a red guy, and blocks. If there's enough of it, it'll be able to block out and force out that intermediate. So now that PABA, which would have led to, you know, essentially down this way, folic acid stuff. And then everything that comes with it, purines, pyrimidines. You remember what these are? These are DNA and RNA. And then amino acids, of course, protein. So they can't do any of that without it. So that's how that works. So I like those are pretty straightforward. So which molecule do sulfonamides disrupt formation of? That was the folic acid. We got this one, right? That's the easy one. Okay, fluoroquinolones. These are DNA or RNA. They're broad spectrum. They're absorbed into the intestine. They can cause seizures and brain disturbances. This is going to include things like CIPRO. If you're interested, there's a breakdown down here. I'm not going to go through it. Next, we have the targeting the cell membranes. We have the polymyxins. These are also present in Neosporin. So these are derived from a bacteria called Bacillus polymyxa, and they destabilize the membranes, and they are toxic to kidneys, unfortunately, but they can be used. And this would be literally polymyxin or daptomyxin. We don't see a lot of that. We have drug-resistant Pseudomonas aeruginosa in urinary tract, and then we have gram-positive bacteria down here with daptomycin. I don't know. I'm not familiar with those drugs. Only just through these slides. So yeah, so Neosporin, we had the bacitracin that inhibits cell wall synthesis. We had neomycin, which inhibits aminoglycoside, which is an aminoglycoside and inhibits protein synthesis. And then we have polymyxins, which inhibit the membrane, right? So that's the trifecta there in Neosporin products. You know, that's why they do so well at preventing infection in any cuts that you get. I mean, I've used, you know, Neosporin my whole life on any cuts or whatever that I've gotten, and it's always never done me wrong. So whatever. So biofilms are hard to treat because they are 1,000 times less sensitive to the same antimicrobials that would be used against like a type of bacteria that would be by itself, like not in a biofilm, because they're all piled up onto one another and with everything stuck together. So we treat them by interrupting quorum sensing pathways. That's important to remember. And we'll also add the DNase to antibiotics to break down the DNA, literally. And we'll like pre-treat somebody that is going to have like a heart valve or something put in that are susceptible to forming biofilms on them, so that, you know, that doesn't happen. Quorum sensing, do you guys remember what that is? That is how the bacteria will like kind of communicate using chemical signals with one another to see like how big the biofilm is getting and like, you know, how things are going around them. So I'm not talking, but, you know, chemical signaling. I sensed that this guy digested this, so that must, whatever. You guys get the picture. That's quorum sensing. Okay, that's important. For treating fungal infections, we have the macrolides, the azoles. That's a pretty big one. You should definitely probably be aware of the azoles. Echinocandins. I mean, these words, nucleoside analogs. Nucleoside analogs literally are things that are similar to the bases in the nitrogenous bases in the DNA and RNA. So it kind of blocks that way. If you're interested in reading about how these work, there is a table over here to the right, but I'm not going to go over it. But common, you know, azoles. Azoles are probably the most common one. This is one I am most familiar with, except for in the macrolides, you have amphotericin. But yeah, you can remember ketoconazole. You should probably be familiar with that, and miconazole. Anyways, that's the fungal ones. The helminths and protozoans. Okay, so these are like our antiparasitics. So we have anti-malarial drugs that are based on quinine. Quinine was derived from a tree bark hundreds of years ago. They've used it for like a long, long time, and we still use it to derive our drugs from, even still today. And then we have metronidazole, which is very commonly used to treat protozoan infections. And they also use it, I was gonna say, also use it for some bacterial infections, the anaerobes. So very interesting, widely used drug, metronidazole. So moving on to the worms, most of these are going to be broad spectrum, and they're used in roundworm intestinal infestations, inhibit the function of microtubules. Remember, that's for moving things around for the cytoskeleton of worms, eggs, and larvae. So that's pretty commonly used in dogs for heartworms and stuff like that. We have the antiviral chemotherapeutic agents. These are, so viruses are difficult because they do hijack our cellular machinery, right? So how do you target the virus without being toxic to the host? So here are some of the major modes of action. These are probably important to know. I would probably know these. Remember how we started off with, in chapter 11, we had the big four. We added on a fifth in this chapter for antibacterials. That was the folate synthesis, right? So for the viruses, these are the three you would want to know. This is going to be preventing penetration of the virus into the host cell. Barring is fine. If you like that word, I don't know. So you want to prevent it from getting in. You want to block transcription and translation of the viral molecules. You don't want it to be able to use any of that. You want to prevent maturation of the viral particles. So these are things that we can prevent and target with drugs. How are we going to do it? So to inhibit the entry, so this was number one. To inhibit the entry, we have enfuviritide and amantadine and zanavir and oseltamivir, which is Tamiflu. And this one is Relenza. I'm sure you can read because, I mean, I just like wrote all over the place over here. Sorry, but you get it. So enfuviritide is HIV. And then these ones all are for influenza. So you see how specific these are. They're usually, not always, but often very specific to specific viruses, because they have to be very specific just to work for that virus, and not harm our cells. So essentially, these are when we talk about Tamiflu. Tamiflu is a pretty good one. I think we're going to see a little slide of it later, but essentially, it's a drug. The flu comes, I mean, it does both entry and also assembly. So I don't know, but it basically affects the neuraminidase, which is a molecule. Like when we say H1N1, right? This is hemagglutinin. Let me write it for you so you can see. And this one is neuraminidase. These are important to know. You don't need to know them right now. These are not going to necessarily be like super important right now, but in unit 4, they're going to be important. Because you're going to need to know it. But neuraminidase is the N, and it's on the outside of the virus, and it sort of allows for interaction with the outside of the cell a little bit more specifically. So we'll just leave it at that for now, so that it will inhibit that action. And it's also important when the virus buds out and comes out, it helps clip it off, so it can free itself. So that's how Tamiflu works. Anyways, these next ones. This is number 2. This one is going to be the nucleic acid synthesis one. So here we have acyclovir. This is probably one of the most important antivirals that we can talk about in this class. So do be familiar with this. There will be questions about this, I swear. So this is a purine analog. So it mimics the bases and it terminates DNA replication. It is developed specifically for herpes viruses. It can be used for other viruses though, but it was developed for herpes viruses. Yeah. I don't know what else to say about it. Okay, so moving on, we also have ribavirin, which is a similar idea. That is a purine analog. We treat RSV. Then these ones down here are the HIV ones. They work a little bit differently. This one does, and this one does, but it's the same idea. They're analogs of bases so that they can get stuck, stick up everything and stop everything from working the way it's supposed to. So that's how it works. Then next we have the things that inhibit viral assembly or release. We have adenavir and saquinavir, and these are also HIV. I don't know much about these, but there's some examples there. So which of these compounds would be best to treat a severe genital herpes outbreak? Do you guys remember? Let's say cyclovir. So that's the one that I want you guys to be familiar with. It's okay if you didn't get it right then, but I do want you guys to try to familiarize with it. Man, my hair just didn't hold the curl today, did it? Drug resistance. So when organisms become resistant to a drug, what is happening? So it's usually an adaptive response where microorganisms begin to tolerate an amount of drug that would ordinarily be inhibitory. How can this happen? This is usually due to genetic versatility and adaptability of microbial populations. Because they are growing so fast and multiplying so much, they can have introduction of, beneficial, mutations, beneficial mutations that are going to help them survive better in that condition. So the ones that do survive better in that condition will survive better and then go on to reproduce. Okay, so we can have intrinsic versus acquired resistance. Okay, so intrinsic is like already in that, right? So they must be resistant to antibiotic that they themselves produce. So that they are already, already made up. You have to, if you produce penicillin, you have to already be resistant to penicillin. That's intrinsic. When we're talking about acquired, this is bacterial resistance to a drug which you were previously sensitive. You had to acquire it somehow. So resistance through spontaneous mutation is common, but in each mutation, there's a minimal chance, very minimal chance that the mutation will be advantageous. There's an even smaller chance that the mutation will confer drug resistance. So it could be advantageous in any way. Now that you want it to be specifically drug resistance, I mean, it's like unlikely. However, large microbial populations and a constant rate of mutation ensure that such mutations do occur. We also have resistance through transfer, horizontal transfer. These are called R factors. These are plasmids that are transferred through conjugation or transformation or transduction. Just plasmids that get from one bacteria into another that carry resistance sequences. These often can occur. We can also see this happening with transposons. Let's just say that. Transposons, those ones that can kind of shift around in locations and that can be conferred from one, from the plasmid to the chromosome or the chromosome to the plasmid and back and forth. So this was what's important for the development of drug-resistant Neisseria gonorrhea, because gonorrhea is becoming increasingly drug-resistant nowadays. So talking about specific mechanisms of drug resistance and how this would work, we have penicillinase, which is an enzyme that's going to break down that beta-lactam ring. Remember that beta-lactam ring? I call it a ring. It's a square, the beta-lactam square. Beta-lactam square ring. It can break that. That's what penicillinase does. So that's how that, if it gets the gene or, has been passed on that gene or a mutation that makes it resistant to that, then that is how that happens. So anyways, moving on. Then we have permeability or uptake of the drug into bacterium is decreased. Sorry, I just can barely see it. I mean, this usually is, from a mutation. So it just doesn't allow the drug to be taken up. So it doesn't get into the cell to do its job. or these ones will just eliminate it. And then these ones, sorry, this is number 4, in case you guys aren't keeping up. These ones will alter themselves so that the thing can't bind as well. So it has a change in the ribosome, for example, in that picture. That makes it so that the drug can't bind as well and inhibit it. So, and that's an example there. Or the drug might act at this pathway, but then the bacteria develops other pathways in order to get to the final product. You might see that with a sulfa. Okay, I hope this one works. Yeah, okay, just click on it over here.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, of course, thin agar that bacteria can move around it. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100.
Oh, I'm sorry.
And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
I always thought that video was really neat because it really puts it into perspective, like how, like when I tell you those were normal wild type, bacteria, they didn't have any resistance at all, and they just naturally developed it from, just being there. So that's pretty neat, I feel like. And this over here, oh gosh, I'm gonna do this over here. So you might have like a little bit of a drug resistant mutation when you have no antibiotics in the broth or this agar. But then if you add antibiotics, you're going to be selecting for it. And then those are going to be the ones that selectively grow up, obviously. So now you have a big growth of ones that are resistant. So that's kind of even how it works in the body as well. So 75% of antimicrobial prescriptions are for throat, sinus, lung, and upper respiratory infections, the majority of which are probably viral and not going to be affected at all by antibiotics. So we're overprescribing is what this is saying. And then we have a shotgun approach to antimicrobial therapy where we're just, we'll just cover everything. And then, you end up getting resistance to those from bacteria that you didn't even think you were going to be affecting. Then we have tons of excess antimicrobial drugs that are produced in the US that are exported to other countries, and that is causing issues as well. Also, the agricultural industry, they're over, antibiotic treating their cattle and stuff like that, and that's causing antibiotic resistance as well. Oh, that's coming up. I didn't realize. So yeah, hospital environment continually exposes pathogens to a large variety of drugs. Of course, it has to. That's what people are there for. They're there to get treated for whatever, disease or whatever they have. So hospitals, they'll house the susceptible patients that already have weakened defenses, and then their workforce may not strictly adhere to the universal precautions, not always on purpose, right? Sometimes you just forgot to, wash or whatever. That's all it takes to carry it over to another room. And then this will lead to penicillin resistance, and that has led to penicillin resistance in nearly 100% of Staph aureus, Staph strains in 30 years. So pretty nuts. So right, this is what I was talking about. 80% of all antibiotics in the US are given to livestock. Supposedly, it decreases infections and improves animal health and size. Enteric bacteria share plasmids that are constantly selected and amplified by exposure to drugs, and these will jump from the cattle to the humans fairly easily, and that will cause spread of drug-resistant infections that way. So these are some drug-resistant bacteria or diseases that we are concerned about at this time, right? So we have urgent down to concerning. So these are the big ones up here at the top are the worst or are the highest concern. And then these ones are the ones that are concerning, but aren't quite as high. So that just like puts it into perspective as you guys are looking through this. Yeah, I don't know. It's a lot. So long-term strategies to control drug resistance include restriction of the use of first-line antibiotics. You don't want to use antibiotics if you don't need them. Enhancing surveillance mechanisms. We have PulseNet and World Health Organization Net, WHONet, that kind of track all of this. And so they'll track foodborne pathogens and pathogenic microbial populations. They can also allow for tracking of resistance and changes in resistance genes, so they can keep track of, you know, what strains might be getting more resistant and where to predict it. So the past approach was finding new targets in the bacterial cell and custom designing drugs against those new targets. But lately they have seen disable. I don't know what disable is supposed to mean. I think it was supposed to say something else. But anyways, host molecules that are used by the pathogens can be like, you know, essentially targeted in order to allow us a different approach to developing new antimicrobial therapies. Some of the low-tech solutions that people have used to try to have a different new approach to this therapy would be using bacteriophages, having bacteriophages that target specific bacteria, and then they kill the bacteria, those specific bacteria, and only those. So those are always specific to specific bacteria. Then incorporating phages into wound dressings themselves. And phages are extremely specific and only affect one species of bacteria, and they leave the normal microbiota alone. So they shouldn't be affecting it unless like your, you know, disease is being caused by E. coli, and then it's going to be causing, you know, issues for all the E. coli. Maybe you have normal E. coli in your belly. I don't know. That could cause some problems, but yeah, you get the idea. So how do we can also help nature, I guess, I don't know how to word that, but you can also help nature along by, you know, introducing probiotics. So these are preparations of live microorganisms that we feed to animals and humans that improve the intestinal biota population. And I took this test online that like you literally send your poop in and they tell you like how many bad bacteria and how many good bacteria, and you're supposed to eat such and such foods to nourish your good bacteria to help your body like work the best that it can. Makes sense. And it is a real thing. And it could work if I had any good bacteria, which I didn't. I had only bad. It's only bad. That was it. So I was just like, well, I guess we're going to switch to probiotics because that's the only choice I have. Anyway, so yeah, that's I do a healthy dose of probiotics every day, and it's definitely helped my GI tract work a little better, but you do you. So yeah, these will replace microbes that are lost during antimicrobial therapy. So you can fill in the gaps of those seats of where where your normal microbiota would be sitting in those seats. When you take an antibiotic, it'll clear all that out. You don't want bad guys to come sit in them. So now we can use these to help fill those seats and replace. So that'll also help out bacteria that are already there as well. We have probiotic yogurt and we have pills and all sorts of stuff like that. So yeah, they're safe, they're effective, and they're useful in treating some food allergies as well because they're helping train your digestive tract, which is true. Okay, prebiotics. These are nutrients that encourage the growth of beneficial microbes in the intestine. It's like food for them, essentially. It's things that they need that help them grow better. So you want to give them the stuff that they like. And so that's what, like, kind of that diet that I was talking about before. They said eat, these specific foods if you want these bacteria, to grow or whatever. And I didn't have any good ones. And so it was just like, well, guess I'll just eat cake. Like, I don't know. But yeah, anyways, yeah. So this just helps the right things grow in the right way in your GI tract. And it helps them keep the bad guys out. If you have recurring C. diff infections, some people have it in their intestines normally. And so when you take an antibiotic, remember C. diff is Clostridium. Clostridium and Bacillus are spore forming, or endospore forming, right? So they are going to outlive pretty much every freaking thing. So when you take your antibiotics to kill out, your whole gut is done except for your C. diff. Your C. diff staying around. So as you're recovering from having taken that antibiotic, C. diff is going to have grown all over and taken over. So that is going to what's causing C. diff infections. Okay, so that's what the overgrowth is being caused by. Anyways, that's how you can understand C. diff and why it works that way and all that. But we can have a transfer of feces, and we can do it. They have pill form. I don't know if you can supposedly order these online. I don't know if it is real. I'm assuming that it is, but I saw it on this video game once about fake dark web stuff. And anyways, it was fake, but I don't know. I'm sure it's real somewhere. But yeah, you can typically, what they'll do is transfer a sample into your gut that's been treated properly through a colonoscopy or an endoscopy, something like that, to help repopulate your gut. So you don't actually have to swallow it and put under for it. Okay, so most common side effect with taking these drugs is diarrhea. That makes sense. We're knocking out all of our normal biome and it's not going to work correctly anymore. And that can lead to intestinal irritation, colitis. It can irritate the intestinal lining, disrupt intestinal microbiota, of course. We also have side effects that include direct damage to the tissues through the toxicity, allergic reactions, or disruption in the balance of the normal biota. So these are different kinds. So these are probably the ones that I would try to be most familiar with. The different ways that they can affect you, either by being toxic to the tissues, or causing allergic reactions, or disrupting the balance of the biome. So it's like the big three there. Okay. All right. Organs that are most commonly affected by these drugs will be the liver, kidneys, GI, cardiovascular system, nervous system, respiratory tract, skin, bones, teeth. It just depends on the drug, really. It's not all of them for every single one, of course. This is an example of someone who had taken tetracycline and then got that gray band on their teeth, like I was telling you guys about. That's from tetracycline. So here are some antimicrobial drugs here on this left side of this column. And then on the right side over here, we have the list of damages that they can cause. So please feel free to look through that on your own. I would recommend it just for like your own, knowledge. I don't know. I think that's smart. All right. An allergy, we're talking about allergies, and we're going to get into exactly what allergies are in a couple of chapters, but we're talking about allergic reactions to the drugs themselves. Just briefly, the drug will be an antigen that stimulates the allergic response. So an antigen is anything the immune system can specifically interact with. Not always bad. So usually bad. Usually it's anything the immune system, will attack. So that's a more important thing. That is what it's doing here. It's looking at the drug as something that it needs to attack. So it's foreign. So the immune system sees it that way. That's what antigen is talking about. The immune system is reacting to it. So that can be provoked by the intact drug molecule or substances that develop in the body's metabolic alteration of the drug. As we process it and get it out of our body, some of that stuff can cause reactions or, whatever. But penicillins and sulfa drugs, of course, sulfas and penicillins are the number one most likely to be allergic to drugs that there are. So we have introduction of broad spectrum antimicrobials to treat infection that will destroy your normal biome, which we've said before, even those that are far removed from the original infection. So often this occurs with women when they get a urinary tract infection, and then they will get a yeast infection as a result, because all that bacteria is gone, and so the yeast can overgrow. Same concept with the C. diff, which is what I was explaining earlier. I don't think I need to go over that again. That sort of situation is called a super infection, where the beneficial resident species are going to be destroyed through antibiotic therapy. And then microbes that were once very few in number, like the C. diff, can now overgrow, or like the yeast can now overgrow because those seats are now empty. That makes sense. So they're no longer getting the protection. So that's a pretty important term to be aware of. All right. So what we talked about in this chapter, chapter 12, selecting your treatments, how the treatments act, the drug classes, even the shapes and sizes and all of that of the drugs, drugs against other pathogens, development of drug resistance, probiotics and prebiotics, and then drug toxicity. It looks like the next one we're going to be talking about infection and disease and what that really means. So that's going to be in chapter 13, and I'm looking forward to it because we're getting into the stuff that I like a little bit better now. I only get a couple chapters in here, but the immune system stuff is my favorite. So I'll see you guys soonish, I guess. And yeah, bye.